biopioneers. Today we'll be talking about Chapter 5, Cellular Respiration. Let's go!
Hello guys, my name is Gunshen Hui and now we are going to discuss about glycolysis. So glycolysis has 10 steps. Glycolysis is occurred in cytoplasm. First, glucose has 6 carbon. As we can see here, got 6 circle. Each circle is stand for 1 carbon. So first, glucose will accept 1 phosphate. So it undergoes the first step, which is phosphorylation. So where is the phosphate gained from? The phosphate is gained from the ATP. ATP will release out 1 phosphate and become ADP. So ATP we can remember is as the T is stand for Tiger, which means that 3, and then the D is stand for Doer, which means that 2. So 3 phosphate is become 2 phosphate. So it already released out 1 phosphate. So the phosphate will accept by the glucose and become glucose 6-phosphate. So this reaction is catalyzed by an enzyme which we call that as hexokinase. So the glucose 6-phosphate is very reactive and unstable. So how to change it become stable? We will undergo isomerization. Isomerization will arrange it and become fructose 6-phosphate. And now it is stable. So for the third step is phosphorylation, which means that the fructose 6-phosphate will gain again 1 phosphate. So where the phosphate gain from is same with the step 1, which is gain for the ATP. The ATP will release out one phosphate and become ADP. So after the fructose 6-phosphate against 1 phosphate again, it will change the name into fructose 1,6-biphosphate. And this reaction is catalyzed by an enzyme and is called as phosphofructose kinase. And then now fructose 1,6-biphosphate is very reactive and unstable. Fructose 1,6-phosphate is very reactive and unstable, and it will go to the step 4, which is splitting of sugar. It will splitting out into 2. One is dihydroxyacetone phosphate, which we'll call as DHAP, and the other one is glyceraldehyde 3-phosphate. So now G3P is stable than DHAP, and DHAP is unstable and reactive. Due to this, DHAP will undergo the step 5, which is isomerization. It will rearrange again and become G3P2. So now we have 2 G3P. So each of the G3P will undergo oxidation, which means that it will release out 2 hydrogen. And then where is the hydrogen goes to? The hydrogen will go to the NAD+, and the NAD+ will accept the hydrogen, become NADH plus H+. Now it will go to phosphorylation, which means that the G3P will accept again phosphate, and the G3P will accept the organic phosphate and become 1,3-bisphosphoglycerate. For the step 7 is substrate level phosphorylation. So it means that one compound which will transfer the phosphate to the ADP. So the 1,3-bisphosphoglycerate, it will release out phosphate, and the phosphate will go to the ADP, and the ADP will accept the phosphate, change into ATP again. So now the 1,3-bisphosphoglycerate, after it release out 1 phosphate, it will change into 3-phosphoglycerate, which is unstable. And now after this reaction, after the ADP accept the phosphate, so now it is already pay off the investment of ATP in step 1 and 3. Due to the 3-phosphoglycerate is unstable and reactive, so we will go to the step 8, which is isomerization. So after we arrange it, we will gain the 2-phosphoglycerate. So for step 9 is removal of molecule water. So now it is already removed 1 molecule of water, and we will gain the phosphoenol pyruvate. And for the last step, which is substrate level phosphorylation, it will also release out 1 phosphate and become pyruvate. So now it's left 3 carbon, and it is called that pyruvate. So where is the phosphate go to? The phosphate also go to the ADP, which will accept by the ADP, and after ADP accept the phosphate, it will back to ATP again. So in the conclusion, we will know that the product of glycolysis is 2 pyruvate, net 2 ATP, and the last one is 2 NADH plus 2H plus. That's all. Thank you.
Okay, so I'll be talking about the Krebs cycle. So you can see the Krebs cycle occurs in the matrix of the mitochondria. So when we talk about the Krebs cycle, you should remember 2 main components, which would be your process and also your molecules. Now, it might seem like a chore to memorize all this because all are new terms and not that familiar with us. So I have an acronym here that you guys can use to memorize and understand the Krebs cycle better. Now, let us see the molecules first. So we have O, C, I, K, S, S, F, and M. Now, the acronym for this would be Orang Cakap, I Kuat Sangat Study Physics Mathematics. So when you memorize this, you can automatically relate to the molecules here. Now, O stands for oxaloacetate, C stands for citrate, I stands for isocitrate, K stands for ketoglutarate, but remember to put an alpha before it, so it becomes alpha ketoglutarate. S is succinyl-CoA, another S will be succinate, so you have like familiar molecules here. And then you have F for fumarate and M for malate. Okay, so now we're done with the molecules. Let us go to the process. And for the process here, this is an acronym on pronunciation. So you'll say chow, so, out. Okay, chow, so, out. Okay, so C-I-O-D-O-D. For chow, you have D on top of the O, but for this so and out, you don't have D. Okay, so C stands for condensation, I stands for isomeration, OD, D stands for carboxylation and oxidation. These two occur at the same time. So I made this as one. And OD, decarboxylation and oxidation, occurring again as one. Then we have S and O. S stands for substrate level phosphorylation. My friend would have explained on that before in glycolysis. So substrate level phosphorylation is a process to form ATP. And after that you have oxidation, and again A, addition of H2O, and O, again oxidation. So you have a lot of oxidation here, and I'll further explain it, relating it into the cycle. I've explained on the molecules beforehand, and now what do? You just arrange these molecules in a cycle pattern. Because as we know, Krebs cycle is a cycle. It starts from point A and it goes back to point A. So a cycle starts from a point and ends at the same point. So you can arrange it in a circle like this. So you see you have oxaloacetate, citrate, isocitrate, alpha glutarate, succinyl-CoA, succinate, fumarate, malate. We have all this in a circular form. Now, after this, you can go on and add your processes. Like I said before, chow, so, out. Okay. So C, condensation, isomeration, I, oxidation, decarboxylation, and you can just go on and use the acronyms to convert one molecule to the other molecule. Now let me explain more on the cycle. Now, on an addition, we have acetyl-CoA, but this molecule is not produced in the Krebs cycle. This came from the link reaction. If you remember, we would have learned a small reaction in between glycolysis and also the Krebs cycle, which is called the link reaction. And in this link reaction, we produce acetyl-CoA, which is a 2-carbon molecule. Now we have oxaloacetate right here. This is a 4-carbon molecule. And when this oxaloacetate, it reacts with this acetyl-CoA to form a citrate molecule. Oxaloacetate is a 4-carbon molecule and acetyl-CoA is a 2-carbon molecule. Therefore, it makes sense that citrate is a 6-carbon molecule, right? So citrate will be formed, and this citrate will be formed. But before that, this oxaloacetate undergoes condensation after reacting with acetyl-CoA, and this condensation means that CoA will be removed. Okay, CoA will be removed. When it reacts with oxaloacetate, CoA is not really needed, so it will be removed by the condensation process. Now, after condensation, you will form citrate, and from citrate you have to form isocitrate, and the process involved would be isomeration. Now you can see the names are quite similar, so you can really remember that it's isomeration. And for isomeration, you would have learned in chemistry, you need water, so water would go in. And after this process, water would not be needed, so water would go out. So therefore, water in and water out. Okay? Then you'll form isocitrate. And we know isomeration, you have the same molecules in the compound, just the rearrangement is different. So isocitrate, it remains as a 6-carbon molecule. Next, from isocitrate to form alpha ketoglutarate, we undergo oxidation and decarboxylation. And when you're in the cellular respiration topic, you know that oxidation always involves NAD+. So in chapter 5, you should remember oxidation is related to either NAD+ or FAD. and that we'll see further on, but now let's focus on NAD+. So what happens here for oxidation process, NAD+ would be turned to NADH. Okay, NAD+ turns to NADH through the oxidation process. Next we have decarboxylation. Decarboxylation means your molecule, it reduces a carbon. So to reduce a carbon, you have to take out a carbon. Okay, you have to take out a carbon. But then you can't just remove it in a form of a pure carbon. What do? You remove it in form of CO2. This CO2 is what you get when you exhale, the end product of respiration. This is the CO2 you're exhaling. Okay, so CO2 will be removed through decarboxylation. Next, we have the same process here. What happens is oxidation. What did I tell earlier? NAD+, would be turned into NADH. Okay, NAD+ will be turned into NADH through this oxidation process. And now decarboxylation, again, you reduce a carbon. So since citrate, you reduce a carbon, you form alpha ketoglutarate, which will become a 5-carbon molecule. So you remove a carbon. From 6 carbon, it becomes a 5-carbon molecule. And when you undergo decarboxylation again, you will form a 4-carbon molecule, which is succinyl-CoA. So how do you remove the carbon? You remove it in the form of carbon dioxide. So you remove it in the form of carbon dioxide. Okay, so now after that you will form succinyl-CoA. Where did the CoA come from? So here you have to add in, you can see we removed a CoA here, right? This CoA will be added back into this process. So there will be CoA added back inside this process. Therefore, you will get succinyl-CoA, which will be a 4-carbon molecule due to the decarboxylation of alpha ketoglutarate, which is a 5-carbon molecule. There will be succinyl-CoA, a 4-carbon molecule. And succinyl-CoA then undergoes SLP. What is SLP? I have explained earlier. SLP stands for substrate level phosphorylation, which is a process to form ATP. So from this, you know that you're going to be forming an ATP molecule right here. But you see, succinyl-CoA is actually a very molecule that's very high in phosphate. So you can't really use NAD+. You can't really form ATP just like that. Okay, you have another molecule helping. So what molecule would that be? It would be GDP. So GDP will be formed to GTP. GDP will form to GTP, and what happens? It receives a phosphate, an organic phosphate from the substrate level phosphorylation. So what happens? It is that it receives a organic phosphate. So GDP will turn into GTP. So what happens here? You have to form, now you can form ATP. Okay? So your ATP will be formed. Where ADP, it turns into ATP. And now you can get succinate. But did you notice that the CoA is no more? That means your CoA is removed from the cycle. Therefore, your coenzyme A will be removed too. Okay, so now you finish your substrate level dephosphorylation. So what happens is you form succinate, which is a 4-carbon molecule. It stays a 4-carbon because there's no decarboxylation processes involved. So it stays a 4-carbon molecule. And now succinate, it undergoes oxidation. Now for this oxidation, you don't use NAD+. For this oxidation, it's quite special. You use FAD. FAD will be converted into FADH2. Okay? FADH2. Oxidation, FAD will be converted into FADH2. Okay. And then what happens is fumarate will undergo the addition of water to form malate. Okay? So this FADH, it will form FADH2. Okay, so fumarate will undergo the oxidation of water to form malate. Okay, malate. Malate is also a 4-carbon molecule. So what happens? It's a very simple process. You just add in H2O, just like that, and just like that you can form a malate molecule. Now, this is the final process of the Krebs cycle. We have come to the end. Malate molecule will undergo oxidation, and here you will use NAD+ back. Okay, so what happens is NAD+ turned into NADH. Okay, so then you can form oxalic acetate, and the whole process or the whole cycle will repeat and repeat and repeat to form ATP for our body. Okay, so yeah, that's the end of the Krebs cycle. Thank you.
Well, I have prepared one question regarding to this glycolysis, so let us give it a try. The first one is glucose, and this glucose will undergo is phosphorylated to form glucose 6-phosphate. And here occurs energy investment, which is phosphorylation. And during this process, it is catalyzed by an enzyme called hexokinase. And later, glucose 6-phosphate will undergo isomerization, and it will rearrange into fructose 6-phosphate. After that, fructose 6-phosphate, and here occurs energy investment again. Which is phosphorylation. This 6-phosphate later will undergo energy investment, which is phosphorylation to form fructose 1,6-biphosphate. And then fructose 1,6-biphosphate will split into two, which is DHAP and also G3P. DHAP will undergo isomerization, and it will rearrange into G3P. G3P later will transfer 2 H atom to NAD+ to form NADH and H+. And G3P later will also combine with inorganic phosphate to form 1,3-biphosphoglycerate. 1,3-biphosphoglycerate later will undergo substrate level phosphorylation, which here also occurs ATP production, and it will form 3-phosphoglycerate. And at last, pyruvate will be formed. So let's move to our question. Okay, so let's start the first question. Label enzyme A, B, and also molecule C. Refer to our diagram. Enzyme A is enzyme A is hexokinase. And the enzyme B, it is phosphofructokinase. And for the molecule C, it is G3P, but we write its complete name. G3P will also call glyceraldehyde 3-phosphate. And the second question, in which part of the cell does this process occur? As we all know, glycolysis it occurs in cytoplasm. And for the third question, what is the net ATP from this process? And the answer is 2 ATP. see here, glucose it changed to glucose 6-phosphate. It is energy investment, which means minus 1 ATP. And here also, fructose 6-phosphate it changed to fructose 1,6-phosphoglycerate, also an energy investment, which also means minus 1 ATP. And here, 1,3-phosphoglycerate changed to 3-phosphoglycerate. It is ATP production, which means plus 2 ATP. And at last, there is a process which 2-PEP will change to 2-pyruvate. And here's a process, it will produce 2 ATP. So minus 1 ATP minus 1 ATP total minus 2 ATP, and here's plus 2 ATP, so equals to 0 ATP. But lastly, 2 ATP, 2 PEP form 2 pyruvate, it will form 2 ATP, so the net ATP is 2 ATP. And for the B question, state the type of ATP formation involved in glycolysis. It is substrate level phosphorylation. And the oxidizing agent, it is NAD+, because it receives electron to form NADH and also H+. And the reducing agent is G2P, because it transfers 2H atom to NAD+. And briefly explain why ATP is a useful substance in living organism. It is because ATP has 3-phosphate group. Together by high energy bonds, and this bond is easily break down also reform. So that's all for the question regarding to glycolysis.
Hi guys, now I would like to discuss on a question, a past year question on Krebs cycle. So we will see the first question, that is name the substances labeled A, B and C. So first we will take a look what is A, B and C. A is the third step of Krebs cycle, where isocitrate undergoes oxidative decarboxylation to become alpha ketoglutarate. So A would be alpha ketoglutarate. Next is B, that is the seventh step in Krebs cycle, where water is used to convert fumarate into malate. So B would be malate. And C refers to the eighth step in Krebs cycle, where malate undergoes it oxidizes into oxaloacetate. So that would be now the second question is state the place where the Krebs cycle occurs in a cell, that is the matrix of mitochondria. So what exactly is the matrix of mitochondria? That is, this is organelle mitochondria, and this is phase A. outer membrane space. And matrix is basically the liquid in this area. So releasing carbon from the substrate, that is Re-Carboxylation. For instance, Re-Carboxylation, it occurs in the third step of Krebs cycle, where alpha ketobutyrate undergoes oxidative decarboxylation to become succinate. And the next question is, state the end products from Krebs cycle. So there are 6 different reactions which forms in ATP, that is, firstly 2-isocitrate converts to 2-alpha ketobutyrate. So the ATP form is 2-NADH. Next is also the ATP form is 2-NADH. The reaction is 2-alpha ketobutyrate becomes 2-succinyl-CoA. Next is 2-succinyl-CoA becomes 2-succinate, and here 2-GTP is formed. Next reaction is 2-succinate converts to 2-fumarate, where 2-FADH2 is formed. And next is 2-malate, it converts to 2-oxaloacetate, where 2-NADH is formed. So you can simplify that as 6-NADH, 2-ATP, 2-GTP becomes 2-ATP, and 2-FADH. So that's all for today.